Module 17, Chapter 27. In this module, we'll be focusing in on aspects of the male reproductive system. However, before starting, I want to just talk about some general characteristics of the reproductive system as a whole. First, this system is involved in sexual reproduction, meaning this system is responsible for the production of offspring by making germ cells called gametes. The female gamete is called an ovum. The male gamete is called a sperm. Now, this system is also unique because it's the only organ system that does have anatomical and physiological differences between male and female. It is also the only system with latent onset. What this means is that this particular system is not fully functional at birth and does not become fully functional until puberty. All of your other organ systems are functional in utero and from there on forward. Also, this is the only system not concerned with individual survival. So you could completely remove all of the reproductive organs from an individual and they'd still live, they'd survive, no problem. Little weird things might happen, but it's not going to cause the individual to die if they don't have a uterus or a prostate. This system, however, is concerned with the survival of the human species. So I guess its functions are a little bit more grand. Before we get into the nitty-gritty of the male and female reproductive systems, there is some information that I want to make sure I go over because I don't know the backgrounds of all the students in the class. Some of you might have heard this information before, some of you haven't. So if you have heard it before, this is a nice review for you. If you haven't heard it before, you'll have some background information to support you when we start talking about how you form your ova and your sperm. Now, the cells of the human body have 46 structures called chromosomes in their nucleus. Chromosomes are the structures composed of DNA so this is where all of your genes are located. And these 46 chromosomes actually exist as 23 pairs. So you have 23 unique types of chromosomes, 2 copies of each. So you have 2 number 1s, 2 number 2s, 2 number 3s, 2 number 4s, 2 number 5s, etc. There is one pair, however, that's not numbered. The pair is referred to as the sex chromosomes. And in females, you'll have 2 X chromosomes. In males, you would have 1 X and 1 Y. Now, because your 46 chromosomes exist as 23 pairs, so you've got 2 copies of each, your body cell, these nuclei are referred to as diploid. So think of di means 2. You have 2 copies of everything. The reason I'm mentioning this is because your gametes are different. An ovum and a sperm, in their nucleus, they don't have 46 chromosomes. They only have 23. So in your gametes, you only have 1 copy of each chromosome. You have 1 number 1, 1 number 2, 1 number 3, 1 number 4. Specifically with the sex chromosomes, you only have 1. Now, for an ovum, the sex chromosome will always be an X, because that's all females have. That's not the case for males, though. A sperm could have an X, or the sperm could have a Y. This is the reason that sperm actually determine the gender of your offspring. So whether your offspring gets the X-containing sperm or the Y-containing sperm will determine whether your baby is a male or a female. Now, because the gamete only has half of the number of chromosomes that all of your other body cells have, the term given to describe a gamete is haploid. So take the HA as meaning you have half of what all of the other body cells have. Now, your gametes are going to form through a process called meiosis. Meiosis is a special type of cell division in which your chromosome number is going to be reduced in half. Now, this is important because new life requires a diploid nucleus to be viable. Viable means survivable. So the new life has to have 46 chromosomes per cell, and in that 46, you have to have 2 copies of each chromosome. So if you have a sperm with 23 chromosomes, 1 copy of each, and 1 egg, which is, we're going to use ova, or ovum, okay, we're going to go with the biological, with 23 chromosomes, 1 copy of each. When that sperm fertilizes that ovum, you have a single cell, which is called a zygote. And this zygote is the beginning of your new life. The zygote now contains 46 chromosomes. It is diploid, 2 copies of each of the 23. So let's talk about the male reproductive system first. Now, most lecture textbooks do the male reproductive system first, with the female reproductive system second. Now, just to set the record straight, the reproductive system is just one system. You have male reproductive structures and female reproductive structures, and then functions that are male and functions that are female. The Saladin textbook is the first book that I have ever seen where it lists the male and female as 2 different systems in 2 different chapters. Most 2-semester books do have just a reproductive system chapter, with the first half being focused on male and the second half being focused on female, and that is more following the anatomical standard. Okay, so just kind of remember that you have one reproductive system with male anatomical and physiological aspects, and then female. Now, with regards to the male aspects of the reproductive system, the reason that it's usually done first is that even though male anatomy is more complex than the female anatomy, male hormonal regulation is more straightforward. So usually they introduce students to the male aspect of the reproductive system first, because the physiology is it facilitates better student understanding, and then you can draw upon that and apply it to the female aspect of the reproductive system. Now, we're not going to do all of the male anatomical structures because that's what lab is for. So we're going to focus in on your testes, because your testes are the site of spermatogenesis. So this is where you are going to give birth to new sperm. Now, the testes are paired oval glands. They are located in the scrotum, so they're actually outside the torso. So they're not within a body cavity. And that's important because sperm production actually has to occur at 3 degrees below average body temp. So in order to form sperm normally, you have to have the testes located outside the torso. With regards to ova production, they require body temperature, which is why the ovaries are kept within the pelvic cavity. Alright, so your testes are going to activate at puberty, and within the testes you have several different types of cells. You've got spermatogenic cells, you've got nurse cells, and you have interstitial endocrine cells. So we're going to talk about the first two first. Within the testes, you are going to have these very highly coiled tubules, which are called seminiferous tubules. It's estimated that you have so many of these tubules and they are so tightly coiled that you can go back and forth across a football field with, like, putting them end to end and straightening them out 3 or 4 times. So there's a lot of tubule length in there. One of the cells that you can find inside the seminiferous tubules are your spermatogenic cells, and these are the cells that are actually going to form your sperm. You then have cells that are called nurse cells. The nurse cells within your seminiferous tubules also go by other names. You might see them referred to as sustentocites or supporting cells. We're going to stick with nurse cells for consistency. Also, ease of spelling. Your nurse cells perform a variety of different functions. For example, they maintain something called your blood-testis barrier. This is equal to your blood-brain barrier, your blood-thymus barrier. It's a tight junction barrier designed to make sure that blood components do not have easy access to lumen components within the seminiferous tubules. There are 2 major reasons for why you would want separation between these 2 areas. One is you need to make sure that the lumen of the seminiferous tubule is kept very stable with regards to its chemical composition. For normal sperm production, you need to make sure that you maintain really high levels of androgens, estrogens, potassium, and amino acids. Also, you want to make sure that your immune response is prevented from killing your sperm. The sperm have only 23 chromosomes, whereas the rest of your body cells contain 46. If your lymphoid system could, let's say, see those sperm, they would actually see that those cells only have 23 chromosomes. They would be recognized as foreign entities, and immune responses would be initiated to systematically destroy them all. So you need to make sure that the sperm are kept isolated just for their own survival. Your nurse cells are going to help support mitosis and meiosis, the 2 different types of cell division. This particular function does require stimulation by follicle-stimulating hormone and testosterone. They help to support spermiogenesis, which is the birth of sperm shape. To do this, they provide nutrients and chemicals that are going to support sperm development, and they help to phagocytize excess cytoplasm that is shed by developing spermatids. Your nurse cells are going to secrete inhibin, which is a hormone that decreases follicle-stimulating hormone production by the anterior lobe of your pituitary, and perhaps has a role on inhibiting gonadotropin-releasing hormone secretion by the hypothalamus, but this is still has not been confirmed. Nurse cells secrete androgen-binding protein, which is abbreviated ABP. ABP binds testosterone and helps to keep its levels high in the seminiferous tubules, which is crucial for spermatogenesis. And the production of ABP is increased by follicle-stimulating hormone. They also produce a secretion called Mullerian inhibiting factor. Now, MIF causes regression of the tissues that would form the uterus and uterine tubes. What's interesting about the reproductive system is that in an embryo, before gender becomes apparent, when the embryo forms, all of the tissues to form all of the reproductive structures of both genders are actually present. And based on your DNA and the hormones that you are programmed to produce, and the different chemicals that you produce, certain tissues will be selected to survive in order to form the anatomical structures of that particular gender. So MIF in males is going to tell the tissues that would form a uterus and uterine tube to basically die, because your embryo is male, you don't want to form a uterus, you don't want to form the uterine tubes. The third type of cell in the testes are called your interstitial endocrine cells. These cells are located outside the seminiferous tubules, and their purpose is to secrete testosterone, which is the primary male androgen. So this is your primary male sex hormone. All right, so now let's talk a little bit about spermatogenesis within your seminiferous tubule. Spermatogenesis is the formation of sperm. It begins at puberty and does not stop with an increase in age. So males have the ability to make sperm throughout their entire lifetime after puberty. However, the amount of sperm formed does decline as you get older. On average, healthy male in their 20s and 30s, it's estimated you can produce approximately 400 million sperm per day. So we're talking large quantities of sperm being produced continually. Now, when you start thinking about spermatogenesis is going to begin with a unique group of cells called spermatogonia. Spermatogonia are stem cells located near the basement membrane or the outer edge of your seminiferous tubule. Spermatogonia are diploid, and these spermatogonia undergo cell division, and when they do, they are going to form two diploid daughter cells. One daughter is called the type A spermatogonia. The type A spermatogonia actually is retained as a stem cell. Okay? If males are going to continue to produce sperm throughout their entire lifetime, you have to make sure you have a constant supply of stem cells throughout the entire lifetime. The other daughter cell is a type B spermatogonium. This cell is going to develop into a cell called a primary spermatocyte, which is still diploid. Now, the primary spermatocyte is going to undergo meiosis I. And in meiosis I, this first round of cell division, your primary spermatocyte, which is diploid, is going to form two daughters, which are called secondary spermatocytes. Now, what's interesting is that your primary spermatocyte, which is diploid, in the figure you'll see diploid represented by 2N, which is the abbreviation for diploid, 2 copies of each chromosome. When it divides and forms its 2 children, they are only haploid. So the secondary spermatocytes only have 23 chromosomes in their nuclei. Now, looking at the figure, when you see that meiosis I and you have your 2 children, the 2 cells that form are roughly the same size. So meiosis I actually involves what is called equal cell division. So when the 2 cells separate, they roughly get the same amount of cytoplasm, so the 2 daughter cells are approximately the same size. Also, I want you to notice that the 2 cells are not completely separated. There's a little teeny tiny connection between the 2. It's called an intercellular bridge. Now, there's a purpose for having this connection. One secondary spermatocyte got the Y chromosome, the other secondary spermatocyte got the X chromosome. And there is a chemical produced by the X chromosome that is needed at this stage for the cell to survive. The Y chromosome does not make this chemical. So this little intercellular bridge allows for this X factor to actually be transported across the cytoplasmic bridge into the Y-containing cell, allowing it to continue to survive. Now, the secondary spermatocytes then immediately begin to undergo the second round of cell division in meiosis, which is called meiosis II. So in meiosis II, each secondary spermatocyte divides, yielding a total of 4 cells, called spermatids. Each spermatid is haploid, contains 23 chromosomes. 2 of the spermatids contain a Y chromosome because they came from the secondary spermatocyte containing the Y. 2 of the spermatids contain the X chromosome because they're coming from the secondary spermatocyte with the X. Now, if you look at those 4 spermatids, they are all roughly the same size. So meiosis II also uses equal cell division. And if you look at the cells, they're still all connected to each other, meaning the Y-containing cells still need that X factor from the X-containing spermatids in order to survive. Now, if you look at those spermatids, they do not in any way look like the mature sperm, which you can see at the top of the field, which means our sperm production is not yet over. Once we have our spermatids, once we have our 4 cells that are all haploid, they are going to undergo a process called spermiogenesis. And this is when your spermatids develop their typical sperm shape and become functional sperm cells. And during spermiogenesis, the cytoplasmic bridges are lost because now the Y-containing cells can survive on their own, and we end up with what is described to be a mature sperm shape. Now, in the sperm shape, one of the structures is called the head, and this is where the nucleus is located. So this is where you have your 23 chromosomes. And on top of the head, sort of like a cap would sit on your head, is a structure called an acrosome. The acrosome is a lysosomal cap. So it's going to contain some digestive enzymes that are going to be important for the process of fertilization. So we're going to use the enzymes within the acrosome to help this sperm penetrate through the walls surrounding your egg, or your ovum, pardon me. Immediately adjacent, connected to your head, is a structure called your tail. The tail is the flagellum, and the tail has 3 different regions to it. You have an area called the middle piece. Now I know the Saladin text calls it the mid piece, but that is not the appropriate term. It is called the middle piece. The middle piece is where you have your mitochondria. Remember, these sperm are going to have to be able to swim through the female reproductive tract to get to an ovum, and that means you're going to have a source of ATP to get that flagellum to move to create propulsion. So the middle piece is where we have your mitochondria. The principal piece and the end piece then work together as sort of the whip-like tail that actually creates the propulsion. Now, the little constricted area between the head and the tail is actually called the neck. The Saladin text doesn't mention the name or label it, but in this neck you have the centriole that the male contributes to the zygote so that it can undergo cell division and actually form that multicelled embryo. Now, once you have that nice mature sperm shape in the seminiferous tubule, that sperm is still not capable of fertilizing an egg, believe it or not. Your sperm still has to undergo one last process, and it's called capacitation. Capacitation occurs in the female reproductive tract. And in capacitation, you're going to see an increase in sperm motility. So those sperm are going to have faster motion of their flagellum. You will also see the plasma membrane become altered on the head, and this altering of the head is going to help prepare the sperm head for fusion with the egg. So the sperm initially can't, if you were to put a sperm from the seminiferous tubule with an ovum in a dish, you're not going to have any fertilization possible. There are chemicals within the female reproductive tract that are going to cause these changes to your sperm. You can, for in vitro fertilization, artificially induce capacitation so that you can have functional sperm penetrate that ovum and yield new life. So, how do we control the functions of your testes? Because if you're producing sperm and you're producing testosterone, you want to make sure that you're doing them appropriately, that you're producing them when you need them, not when you don't. So, the hormonal control actually begins at puberty when your hypothalamus begins to produce gonadotropin-releasing hormone. Now, gonadotropin-releasing hormone, just to recap from your endocrine chapter, travels from the hypothalamus down to the anterior lobe of your pituitary, where it targets gonadotropic cells. And the gonadotropic cells cause a simultaneous production of follicle-stimulating hormone and luteinizing hormone. Now, this is different than in females. In females, the secretion of these two hormones is more consecutive, not simultaneous. Now, when follicle-stimulating hormone and luteinizing hormone are put into the blood, they're going to travel down to the testes, and luteinizing hormone is actually going to stimulate the interstitial endocrine cells. And it's going to cause, by stimulating your interstitial endocrine cells, they begin to secrete testosterone, the most common male androgen. Now, some testosterone is converted into dihydrotestosterone, or DHT, in some target cells throughout the body. Now, with regards to the functions of testosterone and DHT, there are several. Some of the functions are prenatal. Prenatally, there is some androgen production. We'll talk about that later on. Prenatally, testosterone triggers the embryo to follow a male pattern of development. The DHT more specifically stimulates external genitalia development. Another function of testosterone and DHT is to establish and maintain what are called male sexual characteristics. These are things that kind of leap off the page and say, I am male, not female. For example, testosterone increases male muscle and bone mass. It will increase the size of the male larynx, and it will determine the number and location of male adipose deposits. DHT, on the other hand, is responsible for stimulating the characteristic male patterns for facial, pubic, and axillary hair development. It increases sebum production by your sebaceous glands, and it stimulates the growth of your apocrine glands, those lovely apocrine sweat glands you learned about in A&P1. Testosterone and DHT also help to maintain your male accessory glands and organs. They're responsible for the growth of your sex organs at puberty, and then also help to maintain their health and fitness throughout your lifetime. These hormones stimulate metabolism throughout the body. They help to increase your basal metabolic rate. They increase erythropoiesis by increasing erythropoietin production, and they also increase protein production. They affect central nervous system function. Testosterone in very early development programs the hypothalamus for a male reproductive pattern for GnRH production. It has been found a long time ago that GnRH is actually produced in different patterns, whether you're male or female. So it's the same hormone, but the frequency at which it's produced is very different between the genders. These hormones also help to program the male libido, which is the male sex drive. It also helps to program other male sexual behaviors. Lastly, testosterone is needed to help stimulate spermatogenesis and sperm maturation. So your nurse cells need testosterone in order to do what they need to do. Now, at the same time that LH is coming down, you're stimulating testosterone production. FSH is also coming down to the testes, and its job is to stimulate your nurse cells. Now, the nurse cells are going to also, working with testosterone, promote spermatogenesis and spermiogenesis, but your nurse cells also secrete the ABP. And the ABP is going to bind the testosterone and help to keep high levels in the lumen of the seminiferous tubules and the epididymis. Now, if you think about all of that, it was sort of one big on switch, wasn't it? You had the gonadotropin-releasing hormone turning on the anterior lobe of your pituitary. And then you had the LH and the FSH coming down and turning on the cells in the testes. However, you don't want either of these processes to be on all the time, because if it was on all the time, you'd overproduce sperm and create an increase in volume within the testes and the epididymis that could potentially be damaging. You also would continually produce testosterone to the point that you now have way too much. So you've got hypersecretion of testosterone, and that would have adverse effects. So you want to be able to turn these processes off. And the off switch is in the form of negative feedback. So testosterone, as it is doing all of its jobs throughout the body, it does travel in the blood everywhere. So it does end up back up at the hypothalamus. And high levels of testosterone will turn off gonadotropin-releasing hormone production by your hypothalamus. If you lose gonadotropin-releasing hormone, you lose the stimulation of the gonadotropic cells within the anterior lobe. If they're not stimulated, they don't produce their hormones, LH and FSH. Without those hormones, your testes isn't stimulated to function either. So it's basically a full shutdown of all of your testes functions. Now, it might be useful, though, to disconnect the two functions of the testes, to selectively be able to turn off your hormone production, be able to selectively turn off the sperm production, but still allow the hormone production. So you do have a dual control mechanism, and that's where the inhibin from your nurse cells comes in. Inhibin selectively suppresses FSH production. So you do have the ability to have inhibin go up to the anterior lobe and selectively turn off just FSH production. Losing the FSH production would decrease sperm production, but you would still be allowing for luteinizing hormone to come down and stimulate testosterone production if you still needed to produce that hormone. So you do have the ability to disconnect the two functions so that you can still produce the hormone even if you don't need to produce the sperm. The semen ejaculated by the male is a solution containing both sperm and seminal fluid. With regards to the sperm component, a healthy male, 20s and 30s, contains about 50 to 120 milliliters of sperm per milliliter. Infertility is usually diagnosed when there's lower than 20 to 25 million per milliliter. The seminal fluid, the fluid portion of your semen, is contributed by secretions from your nurse cells, seminiferous tubules, seminal vesicles, prostate, and bulbourethral glands. Now, if you were to take a sample of semen and look at its components and where do they come from, 10% of your semen volume is composed of sperm and the secretions from your nurse cells and seminiferous tubules. Approximately 60% of your semen volume comes from the seminal vesicles. Approximately 30% of your volume comes from the prostate, and the bulbourethral glands only contributes a trace amount of fluid to your overall semen volume. With regards to the functions of your seminal fluid, this fluid is your transportation medium. Again, your sperm are going to be traveling through seminiferous tubules, through an epididymis, through the ductus deferens, through an ejaculatory duct, and through the urethra in order to exit the male. And what's going to be easier to move through all of those tubes? Something solid or something fluid. So just for general transportation, your seminal fluid's very useful. Your seminal fluid contains the nutrients that your sperm are going to need in order to produce ATP to swim. The nutrient that is present is called fructose, and it's contributed by your seminal vesicles. Your seminal fluid provides protection from the acidic environment in both the male's urethra and the female's vagina. Sperm are very sensitive to pH, and an acidic pH would actually kill sperm. So the bulbourethral glands are going to release their secretion prior to ejaculation. Their secretion is very alkaline, and what it will do is it will neutralize any acidic drops of urine still in the urethra prior to ejaculation. The secretions from the prostate and the seminal vesicles, also very alkaline, and it's going to help to neutralize the acidity within the female's vagina to help protect the sperm from exposure to the acid there. The acidity for the female is essential, though, as a protective mechanism to help prevent pathogen entry. Also, the alkaline environment does help to activate sperm swimming. So it creates the right chemical environment to help activate that swimming motion for eventual migration to the egg. Your seminal fluid is going to help your sperm to be retained in the vaginal canal after it is deposited there. Clotting enzymes from your prostate activate a chemical called prosemenogelin. Sorry, even I have trouble pronouncing terms every once in a while. Prosemenogelin from your seminal vesicles into semenogelin. Semenogelin is actually fibrin-like. So it's more of that mesh-like material that was used to help form your blood clot. And the semenogelin helps to, I guess, make your semen more sticky and enable it to be retained on the walls of the vaginal canal. And by sticking to the walls of the vaginal canal for a short period of time, you offer your sperm that time to actually migrate from the vaginal canal through the cervical canal and into your uterine cavity. You also have chemicals in here called prostaglandins, which come from the seminal vesicles, that help to cause contractions of the female reproductive organs. Now, the movements are described as reverse peristalsis. They are the same rhythmic contractions that you learned about in the digestive system, but instead of the contractions trying to push something out of the body, these contractions happen in reverse to help draw the sperm into the female reproductive tract. The prostaglandins might also help with thinning your cervical mucus so that the sperm have an easier time passing through the cervical canal. There's also an antibiotic chemical in the seminal fluid. It's called seminal plasmin. It's produced by the prostate gland, and it does help to destroy bacteria that might pose potential harm to the sperm.